Aloha everyone, my name is Ciara Ratum. Today I'm going to be talking about my undergraduate capstone project. Objectives of this project include using Flowcam imaging as a method to explore phytoplankton communities within Heʻeia fish pond, also to develop a method of processing these images. Questions pertaining to this study include to see whether there are trends in location, time, season, and environmental conditions for these phytoplankton communities. Currently in Hawaii, we face challenges in food insecurity, climate change, and invasive species. However, there are multiple organizations, including Paipai o Heʻeia and the Heʻeia Natural Estuarine Research Reserve, who are focused on restoration efforts to combat these issues. These two organizations are focused on the restoration of Heʻeia fish pond. And Heʻeia fish pond is a local kuopā style fish pond, meaning it has a wall that encompasses the entire fish pond at 1.3 miles. It is the longest in the Hawaiian Islands. It also holds about 88 acres of brackish water. Within a fish pond, nutrients is the main driver for productivity. Heʻeia fish pond has two main inputs, including Kaneohe Bay and Heʻeia Stream. And this mixing of seawater and freshwater creates an ideal environment for the cultivation of micro and macro algae, which are basically phytoplankton and diatoms. The goal for the fish pond is to cultivate native herbivorous fish, including the striped mullet, ama, and the milkfish, aba. These two fish are known to have diets primarily composing of microalgae or diatoms. And it is crucial to understand the nutrient and environmental factors that promote the growth of these diatoms within their diet to then cultivate these fish. With the restoration efforts, there have been descriptive phytoplankton studies that have been conducted in Heʻeia fish pond and Kaneohe Bay using genomics and data sequencing. However, there are limitations to these studies just due to the availability of DNA databases. So hopefully this alternative method of using the Flowcam can help fill this gap of information. Sampling sites for my study included Waitu, which is the only functioning freshwater makaha, and Kahokele, which is an ocean break makaha. Samples were taken and pre-filtered using a 25 and 54 stacked micron sieve. Those contents were then transferred into a smaller container, which was preserved and then sent to the lab for analysis using the Flowcam. So what the Flowcam is, it is an integrated system that uses microscopy and flow cytometry. It basically runs the sample through a flow cell, and then a camera automatically takes pictures of all of the particles that pass through. And basically what we get back is that screenshot with all the cool little guys. However, it doesn't look as pretty as that picture. There's usually a lot of debris within estuarine samples, especially due to the nutrients. But you're able to use the program to automatically sort it. To do this, you need to create libraries of specific images. A filter can then be generated from that library with its statistics of different variables, morphological variables such as area, diameter, and even symmetry. And you can apply that filter to different samples, which it will then sort out automatically based on that filter. Environmental parameters at makahas were different in salinity and dissolved oxygen. When looking at average diatom density, Phy2 did have chain-forming diatoms vary in relation to season, from wet to dry season. In relation to salinity, we did not see any significance from sampling sites. However, at Phy2, we did see centric and chain-forming diatoms being different throughout sampling dates. as well as Kahokele, we saw pennant diatoms being different throughout sampling dates. When looking at diatoms of interest for Ama Ama and Ava, we based off of findings from previous studies that looked at the gut contents of these fish, we saw that these taxa did vary between dates as well as sampling sites. Some takeaway points include salinity being one of the determining factors for diatom abundance. We did see diatom density vary over different times of the year as well as between makahas. We did see that at Kahokele, diatom density was greater compared to Vai2, which was interesting because with the lack of salinity at Vai2, you would expect there to be more nutrients and productivity. But this could be a factor of competition with zooplankton, it being so rich in nutrients that zooplankton are out competing phytoplankton, and that is something to look at for future studies as well. With the use of Flowcam, hopefully we will be able to fill a gap of information and put this knowledge towards the tracking of these estuarine and marine food webs. Here are my references, and thank you all for listening.